and then this information travels to the hippocampus, where it is then implemented as a memory. So the hippocampus is also responsible for the conversion of short-term to long-term memory. And so many memory-related illnesses or brain injuries cause damage to the hippocampus because it is responsible for memory function. The next thing that I'm going to talk about are neurons. Neurons are how the brain sends information to the rest of the body and receives information from the rest of the body. The way it works is that in order for information to travel down a neuron and then to the next neuron is through the use of electrical impulses, and these are known as action potentials. So action potentials work on a property called a all or nothing property. And so that means that there's a threshold, and this is a graph of an action potential right here. And so as long as the action potential reaches that threshold of that certain membrane potential or membrane voltage, then information will be able to travel from one part of the neuron to the next. And these electrical impulses happen all the way down the neuron until it reaches the end, so that information can travel from that neuron to the next one. So like I said before, if the threshold is not reached, the neuron will not fire. So this is really important for the rest of the paper, so I just want to know if there are any questions, just to make sure everybody understands this part. Awesome. So since we don't know everything about the brain, the scientists who were studying this prosthesis needed to make some assumptions about how the brain worked. So they assumed that the majority of information in the brain is coded by spatiotemporal activity and its patterns. And this basically means that the amount of space in between action potentials codes for different types of information. So like I said before, since it does not matter how high the membrane potential or how high the membrane voltage is, as long as the threshold is reached, it doesn't matter how high up or the height of the action potential, but so they said that it matters about the space in between the action potential, which would cause for different types of information. So like these two action potentials right here, there's a lot more space in between these two than there is between these two action potentials, and so then there would be different information that would be coded for. Long-term memory, I was kind of surprised when I read this. Long-term memory is anything after 30 seconds. Short-term memory is 15 to 30 seconds. And so memories in general are stored as groups of neurons that will fire in the same pattern as the initial event that they were made when you need to retrieve them or call on them. So when you make a memory, just like now, hopefully you guys will remember my presentation forever, there are neurons grouping together, and so they're firing in a certain way. And so when you want to remember my presentation 10 years later, when you call on that, those neurons will fire in that same exact way as they are right now when you're listening to me talk. Additionally, this made me think, why do we forget then if we can all these neurons group together? Well, sometimes, like I said before, the hippocampus is connected to the neocortex. If you're like rushing, say you're like, I forget my keys, or like I lose my glasses all the time. That's mostly because I just throw them around, I'm in a rush, I'm trying to get to the next place. So I'm not really engaging all of my senses. So I don't remember what the couch looked like, what the fabric felt like when I threw it on the couch. So then I'm more likely to forget that because I'm not engaging all of my senses. So then that goes for the same reasoning for why we remember big events like weddings or birthdays, because we're really engaging our senses, we're in the moment. So back to the prosthesis. The location of it is really important. This gray thing right here, that's the prosthesis. And this is the hippocampus, and as you can see, the X represents the damaged hippocampus. So everything that comes into the hippocampus, it'll actually go around and it'll go up into the prosthesis. And so everything that also goes out of the prosthesis will come out and travel in the same direction that it would if the hippocampus was functional. So this prosthesis by any means does not fix the hippocampus, does not replace the hippocampus, but it sort of circumvents it so that it's just, you don't even need to use it altogether. So if you remember the title of the paper, it's through the use of multi-input, multi-output modeling. And so the reasoning for this is because since action potentials, they need to reach a certain threshold in order to allow information to travel down the neuron, sometimes they do not reach the threshold. So you can see in this picture right here, this action potential did reach the threshold and it did allow information to travel down the neuron, but this one didn't. And so this happens a lot of times with single pulses, where a lot of times it will reach the threshold, but a lot of times it will not. This is the same thing with the dual pulses. Most times one will reach the threshold, one won't. Same thing with triple pulses. So they said, well, if we use multiple pulses, multiple inputs, then that will allow us to have more accurate information that comes out of the prosthesis. So you can see through the multiple pulses, even though some of them do not reach the threshold, many of them do, and since some of them do not reach the threshold, that will cause for different spacing in between each of the spike trains, and which I said before, different spacing in between each of the action potentials causes for different types of information. So is everybody good on that? Awesome. So the prosthesis is based off of some really, really complicated math. And if you have any questions at the end, I definitely know how this works. So you guys can ask me some questions about it. But in a more simple term, since these, like I said before, when you need to make a memory, the neurons will group together, and when you call on it, they'll fire in the same way that they did when the memory was made. Well, when your hippocampus is damaged and when your memory function isn't really working, then the neurons that are grouped together are not actually grouped together, and so they won't be able to fire in that same exact pattern. So the prosthesis works in a way like a regression model or a best fit line, where you have all these different data points, and it sort of makes a best fit. So rather than something like this line that doesn't really account for a lot of the data points, through all of these different functions, and mostly this one here, it is able to create a line like those blue, green, and orange curves that best fit the data points or best fit the spike trains that are sort of all over the place since the neurons aren't grouped together anymore. Does that make sense for everybody?
Yes.
Awesome. So this is a more simple way of showing that math. A lot of the variables that were in the math equations before, they're shown here. And so the multi-input, multi-output model, the MIMO model, is actually composed of 3 different MISO models, or multiple input, single output models. So the way these work is all these different spike trains from all these different neurons that are scattered since they're not able to be grouped together. All of those different spike trains will come into the prosthesis, and that's like in that first picture of where the location is, where all of the inputs come in that normally go into the hippocampus. They'll all come into the prosthesis. They'll go through all of those functions where it tries to create that best fit line, and then those will come out as the outputs. And then, so that's the same thing for all the other 2 MISO models, so that you have the outputs that look as similar to what they would be if the hippocampus was actually working, and if the neurons were actually grouped together and firing in that pattern as the initial event. So this is a light imaging technique just used to show the accuracy of the model. So this is a picture. First, the actual is actual hippocampal function where it's not dead at all. The next one is the predicted function, which is predicted by the MIMO model. So as you can see, there's some differences like here and here, but for the most part, they really look relatively the same. So the next thing that they did was experiment on mice. So the way they did this experiment to test to see how well this MIMO model was working was they used a delayed non-match sample task. And so this is a test used a lot of times in animals in order to test their long-term memory function. So first off, they medically blocked the hippocampus so that the hippocampus would be working in the same way as someone who has a damaged hippocampus and does not have long-term memory ability. And so what they did and what's shown in this picture of the monkey is first they're shown a sample stimulus and a reward. So that's the key, and then when the monkey picks up the key, in this case, when the mouse chooses the key, they'll find that there's a reward underneath it, which is most times food. And then there's a variable delay, and so the variable delay increases over time so that it goes from short-term to long-term memory. And then they do a bunch of different trials where instead, even though the key is the familiar object, they then pair the food, the reward, with an alternative object. So they'll switch out that alternative object, but they'll keep the key the same. So if the animal was to choose the key, then that would be testing short-term memory because that's the item that they're familiar with. But since they're testing long-term memory, they want to train them to pick the alternative object, which they will now hopefully have learned is what contains the food underneath it. So the results of these tests, the green line represents a completely normal, completely working hippocampus. The red line represents a knocked out hippocampus, but with the MIMO model implanted in the brain. And then the blue line represents a completely knocked out hippocampus, nothing done to it, just non-functioning. So the x-axis represents the variable delay that's shown here in the picture. And so the variable delay increases over time. And so even though I said that short-term memory is 15 to 30 seconds, and it seems like, well, this is all short-term memory, since the test takes sort of a long time, especially those training sessions where you're switching out the alternative objects, it will actually span into long-term memory. And then the y-axis represents the mean percent correct, which is the amount of times that the animals chose the alternative object which had the reward under it. So as you can see, the control, which is the normally functioning hippocampus, it is very, very similar to the MIMO model, and the MIMO model is definitely working a lot better than a normally non-functioning hippocampus. So even though it's not perfect, and it can't really be perfect right now since a best fit line doesn't always get all of the data points, it is really, really close to it. It never goes below 50%, which is sort of random, like the normally knocked out hippocampus does. So does that graph make sense to everybody? Okay, so with that, the conclusions of my paper, first of all, the prosthesis has real-time learning and it's one size fits all. So a lot of times with other prosthetics, like an arm or a leg, there has to be a lot of rehabilitation done in order to be able to use it, sometimes taking months at a time. But for this prosthesis, it's able to take all of the neurons and the action potentials that are already there, so there doesn't need to be any sort of training with this or modifications for different people. Additionally, I want to stress that it does not fix the hippocampus. It just sort of circumvents it so that it doesn't even need to be used. So it is not a solution for actual hippocampal damage, fixing the hippocampus, but it just totally knocks out the use of it altogether. So some drawbacks or unanswered questions that the paper sort of left up to our thinking was the cost, or can this prosthesis be mass produced? So it didn't really mention that, which is something that's really important if this actually wants to be implemented in hospitals or in military situations. It also didn't say how you could implant this, or which also how invasive it is to implant. If somebody does not have long-term memory ability, but this prosthesis is a working function for it, but then it comes into how much does the surgery cost, how risky is the surgery. So the paper didn't really go into that. And then it also didn't go into how much it extends long-term memory by, just because they only did sort of a few minutes in the animals. It didn't really go into, years and years and years, and so that really can only be answered with the use of human trials. But it's still really, really cool, really innovative, and it was shown to be pretty accurate. So this could be used in military situations. Some statistics are that temporal lobe damage related to memory is common in military personnel in war zones. Over 50% of injuries sustained in combat are the result of explosive munitions. 60% of those surviving blasts sustain significant brain injury, most often to the hippocampus. So this could be something used, in the military, which would be really, really awesome since those statistics are pretty big numbers. Additionally, it could be a solution to memory loss related illnesses, and then it could also be used just in hospitals. With that, thank you guys. I hope you understood everything. I hope you enjoyed my presentation. Thank you to the Journal Club team. I hope you guys enjoyed the research exhibit. Are there any questions? Awesome.
So how do you keep the implant running? How does it get powered?
Well, the paper didn't really go into that, so I can get back to you on that later.
Battery then. There must be battery.
Right.
Yeah.
Any other questions?
How do you plan to take this forward? What's next for you with this?
For me? Well, I'll have to see what the researchers come out with their next paper. This was really recent. This just came out in 2018. So there definitely needs to be more testing. And so, I can't really go into the labs or whatever. But I'll definitely be looking out for that next paper if they do clinical or human trials, or if this can even be implemented, in the real world.
So they did the mouse trial and the monkey trial.
So they did not do monkeys. The picture of the monkeys was just something that I found on the internet just to show. It was just to show sort of how the delayed non-match sample task works. Just because it's sort of a confusing memory test. So I just thought the picture would be a little more helpful to understand it. But they just did this in only mice.
We were wondering where the mice were.
Yeah, no, there are no pictures on Google with mice. So.
How do you choose topics specifically?
Well, I am pretty interested in the brain. Last year I did do a Journal Club presentation also on the brain, about ALS and how, the brain, through the use of a computer, they were able to sort of read their minds. That's a really conflated way of saying it. But I am sort of interested in that, and I also wanted to do something that was really innovative, never been done before. And so I kind of just looked up really innovative, never been done before papers. And it was in some, really cool medical, medical science articles. So that's how I chose this paper.
Did the paper or research cover anything about prior memory? Because the process after you've implanted is from here on out and how it will function. And what happens to, although the hippocampus damage, like memory that was put into it prior to the implant, how does that function?
So it didn't, that's a really awesome question, but it didn't really go into that. But I would assume that since the prosthesis is able to take all the different inputs that are already there.
So it can work in conjunction.
Any other questions? Awesome. Thank you so much.